If you are here, you might have encountered this equation of anion gap at least once in your life. This concept of anion gap is very important clinically as well as from exam point of view. But unfortunately, it is very confusingly presented in our textbooks. In today's lecture, I am gonna demystify the whole concept of anion gap and I will present you in quite palatable form. I will tell you in a logical way that from where this equation of anion gap came, and then we will discuss that in what conditions anion gap is increased or decreased, and how it happens, that why in some condition anion gap is decreased or increased. In a very logical fashion we will see all this, right? So let's start the lecture with a very basic question, and the question is that whether plasma has a negative charge, positive charge, or is it electrically neutral? Yeah, it is electrically neutral, right? So it means that because plasma or blood is electrically neutral, so in order to maintain the electrical neutrality, negatively charged ions in our plasma must be equal to positively charged ions. All sum of positively charged and negatively charged ion must be equal, right? So let's suppose this is the anode, the positive pole of electrical field, and this is cathode, negative pole of negative terminal of electrical field. And if we pass the electrical current through these ions, the negatively charged ions, they will attract it towards positively charged anode. Negatively charged ion will move toward anode and positively charged ions will move towards cathode, right? So because of this reason, as they say, the opposite attracts, right? So because of this reason, these negatively charged anions in reference to anode, they are named as anions, and these positively charged ions in reference to cathode, they are called cations, right? So we can call negatively charged ions as anions and positively charged ions as cations, and they must be equal, right? So this bar graph represents total anions as well as total cations. So total anions and total cations in your plasma must be equal. Clear up till now? So total anions, they consist of those anions that are measured, that are routinely measured in laboratory, and it also consists of unmeasured anions that are not routinely measured in laboratory. So it is sum of total cations is sum of measured anions as well as unmeasured anions. Similarly, total cations is also sum of measured cations as well as unmeasured cations. So in this diagram, total anions is equal to unmeasured anions as well as measured anions, right? Similarly here, total cations is equal to unmeasured cations, which are very small, and measured cations, right? Now what we will do is that from both sides of this equation we will subtract unmeasured anions. So unmeasured anions are subtracted, unmeasured cations are subtracted from both sides of this equation, right? And both of these will cancel each other and our equation will become like this, right? Clear? So next I am gonna rewrite, I have rewritten this equation, and now we will subtract measured anions from both sides of equation. We will subtract measured anions from both sides of equation, and while doing so, these measured anions, both of these will cancel each other due to difference in arithmetic sign. It is positive and it is negative, right? So now our equation will become like this: unmeasured anions minus unmeasured cations, right? Clear up till now? Now let's substitute. We don't know what are unmeasured anions. Of course I will tell you later, but just consider that for now we don't know what are unmeasured anions and what are unmeasured cations. But for measured cations we can substitute with the actual ions and measured anions as well. What are measured cations? These are sodium and potassium, right? Here sodium and very small amount potassium will be there, right? It is very small that I have just ignored it in this diagram. Along with that, measured anions, which are bicarbonate and chloride, right? So measured anions, these are bicarbonate and chloride, and measured cation is sodium. Clear? So when we will simplify this, and when we will remove this bracket, this plus sign will become minus, right? So this is the final equation, and if you look carefully, this is actually the equation of anion gap. So now you understood that what is anion gap, how this equation came from. Basically this equation is just a simplification of this equation, right? It is a rearranging. If we rearrange this equation or this equation, if we rearrange this equation, we will ultimately get this equation. Now you don't need to memorize whole this derivation, this is just for concept, but at least you must understand that what anion gap actually implies, what it actually means, right? So this potassium, some authors, some books simply ignore this potassium, and this equation become like this: sodium minus bicarbonate minus chloride. Yes, you have a question? Oh, that's a good question. He is asking that why there is some injustice with potassium, why they are ignoring the potassium. Very simple. First of all, potassium is very low, so we can simply ignore it. It does not have much impact on this calculation. Secondly, the potassium is not much significant in all this calculation. We will later on come to know that it is not much significant here in discussion of anion gap, right? So we can ignore potassium. Secondly, unmeasured cations are also very small, right? So we can also ignore them, especially while discussing anion gap in respect to acid base balance. We can ignore unmeasured cations, right? Especially when discussing in respect to acid base balance, while in other cases we cannot ignore it. We will discuss that later in detail. So if we ignore this part, this is very small unmeasured cations, this equation becomes like this, right? Clear? So what is the definition of anion gap? Now by now you must have understood. So anion gap is mathematical difference between unmeasured anions and unmeasured cations present in plasma, right? What is anion gap? It is mathematical difference between unmeasured anions and unmeasured cations in our plasma. This is one type of definition. The other definition which some other authors mention, that is anion gap is a mathematical calculation of unmeasured anions in our plasma, right? Because they are ignoring this unmeasured cation, so they simply say that anion gap is mathematical calculation of unmeasured anions in our plasma, right? But why do we want to calculate, why do we want to estimate the concentration of unmeasured anions in our plasma? This is very important concept which I am gonna discuss just now, right? But before that, let's solve this equation. And by the way, this is anion gap if you want to see in this diagram. If we put the values, sodium is, let's say, on an average 140, potassium is 4, and all these values I have substituted, we will get the answer. All these values are in milligram per deciliter, and we will get 12 mg per dl. So basically anion gap is a range, and it varies from 8 to 18, somewhere between 8 to 18 milligram per deciliter. This value you don't need to, probably I don't think you need to remember these values because it differs as per the laboratory and whether or not they are considering potassium in the calculation, right? So this was the equation of anion gap. Now what do I mean by unmeasured anions? What are these unmeasured anions? Yes, what do you think? What could be these unmeasured anions? Yeah, lactate can be there. Other than that, what else? Very simple. Negatively charged plasma proteins. We have many proteins including albumin that is negatively charged. Why it is negatively charged? Because it is having some terminals, some amino terminals that are negatively charged. That is very easy to understand, right? I won't go into details. So negatively charged plasma proteins, for example, albumin. Then we have phosphates and sulfate, PO4 negative and SO4 negative. You know, phosphates and sulfates. ATP have phosphates and sulfate. Glucose, when we in glycolysis we attach glucose 6-phosphate. So phosphate is widely present in our plasma, right? Not widely, it is not too much, but of course it is there significantly. It is present, and sulfates also, these are present in plasma, right? And conjugate bases of organic acid. Now what I mean by this conjugate base of organic acid, just wait for now, I will explain it later, right? And by the way, I have created this Facebook group. Here we have medical discussion. If you are a medical student or pharmacist or paramedical student or doctor or whatever, you are most welcome to join this group. The notes of this lecture and any other lecture that I upload, they will be available on this group. I will share the link in description, right? So now that you have clearly understood that where this equation of anion gap came from, now we will discuss that how in certain condition anion gap is increased, right? So let's suppose these are the bicarbonate ions, and these are bicarbonate is alkali reserve of your body, of your plasma, right? Now let's suppose here is a cell. You know, from glucose you break down the glucose through process of glycolysis and convert it into pyruvic acid, and in this process you get energy in the form of ATP, and then this pyruvic acid is funneled into TCA cycle or citric acid cycle, which is oxygen dependent process, and in through TCA cycle you get some more energy in the form of ATP, right? But let's suppose there is a situation that you are getting less oxygen. Energy demand is very high, but oxygen is low. So what will happen? Due to low oxygen, this TCA cycle, pyruvic acid won't be much funneled into TCA cycle, and TCA cycle will slow down, right? So pyruvic acid will start to accumulate inside the cell, right? And when pyruvic acid will accumulate inside the cell, it will be converted into lactic acid. There are number of conditions that can lead to such situation. For example, let's suppose you are doing a strenuous exercise for a long duration, then lactic acid can accumulate. Similarly, in shock or in cardiac failure, in such condition in which blood is not coming to the cells or oxygen is not coming, for example in respiratory failure, lactic acid may accumulate. So lactic acid, when it will accumulate in our blood, this condition is called lactic acidosis. Lactic acidosis. So what lactic acid will do? Lactic acid, basically lactic acid consists of lactate, which is conjugate base, as well as proton. So negatively charged lactate is attached with positively charged proton, and whole this structure is called lactic acid, right? So in plasma it will be dissociated into lactate, which is conjugate base, negatively charged, as well as free proton. Now this free proton is the actual culprit. This free proton is actual culprit which decreases the pH of plasma. This proton is the chemical that imparts acidity in a substance, right? We have discussed this detail in a lecture of concept of pH, right? So what this proton will be doing, because this proton can decrease the pH quickly, so these bicarbonate, they will come into action and they will fight with this proton. They will neutralize this proton. Of course, we have more and more lactate and more proton. We are not just having single lactate and single proton. So proton will react with bicarbonate, and bicarbonate will be lost, and this thing will be converted into H2CO3. In this way it will neutralize the acid, right? Similarly here it will neutralize the bicarbonate, as well as H positive, and we will form H2CO3, right? You have a question? Yes. Yeah.
But how does it neutralizes the plasma? It is also an acid.
Yeah, very good question. So she is asking that H2CO3, carbonic acid, is itself an acid. So how it can neutralize the acid? The answer is very simple. Actually, it is a quite complex topic, that how buffer work. I will have a separate video on this. But for now, you just understand that putting in H positive is like putting a strong acid in it, right? Putting a very strong acid in it. It will pick up, bicarbonate will pick up H positive and it will convert it into very weak acid. Carbonic acid is very weak acid. So you just consider that a strong acid is being converted into a very weak acid, and because of it acidosis is there, but very strong decrease in pH, very large decrease in pH is not there, right? So you got the point. So pH changes minimize, and that's how buffer work, right? So now what you see here is that bicarbonate ion is continuously decreasing. As more and more lactic acid is being produced, bicarbonate is continuously decreasing. Bicarbonate is being converted into H2CO3, which is a neutral substance. Bicarbonate is negatively charged substance. It is anion. But H2CO3 is neither anion nor cation. It is a neutral substance. So it means we are losing anions. So what will happen? So you see this diagram. Bicarbonate is decreasing. Now because bicarbonate is decreasing and cation, sodium and unmeasured cations, these are same. So what will happen? Plasma will no more be neutral. These positive charge will overpower. So positivity will ensue. Of course that is not possible and that is not intended, right? We must maintain, rather our bodies must maintain electrical neutrality. So how it can maintain electrical neutrality? The good thing is that body does not have to do anything for maintaining electrical neutrality, because we have already got these packaged lactate. These are lactate. Do we routinely measure lactate? No. So lactate is unmeasured anion. So lactate are there to increase unmeasured anions, right? So because of press, yes, you have a question? Yeah, to increase the negativity, and these are unmeasured anions that are increasing the negativity. So what you see in this diagram, that conjugate base of lactic acid has increased the anion gap. Anion gap by definition is equal to unmeasured anion. So unmeasured anion has been increased, right? So conjugate basis of organic acid is increased. Yes, what are you saying? Yeah, excellent. So he is saying that one negative charge is lost in the form of bicarbonate, then along with that we are getting negative charge in the form of lactates. So and we don't measure that negative charge. So because of that we have no change in electrical neutrality. And because of this, this type of metabolic acidosis due to lactate or due to any conjugate base of organic acid, it is called increased anion gap metabolic acidosis. Clear? So one of the causes lactic acidosis, in which lactate is the unmeasured anion and proton will decrease bicarbonate. Another example, salicylic acid poisoning or aspirin poisoning. Salicylic acid, it consists of not salicylic acid, basically it is acetyl salicylic acid. I have wrongly written here, sorry about that. So acetyl salicylic acid, again it will be dissociated into acetyl salicylate, which is negatively charged unmeasured anion. Along with that, this proton, it will decrease bicarbonate. So due to this conjugate base of organic acid, anion gap will increase, and due to these protons, metabolic acidosis will be there. So this will be increased anion gap metabolic acidosis. Clear? Yes. Yeah, he is very right. So he is saying that he has read somewhere that acetyl salicylic acid or aspirin, it can cause respiratory alkalosis. He is very right. Initially it caused respiratory alkalosis. It actually causes mixed type of acid base disturbance, right? Initially it caused respiratory alkalosis because it increases respiratory rate, which leads to wash out of carbon dioxide. I'm not gonna go into details right now. So it will lead to respiratory alkalosis, but when it will go into blood, it will also decrease bicarbonate through metabolism. It will also decrease bicarbonate, and there will be increased anion gap as well. So there will be respiratory alkalosis plus metabolic acidosis. Mixed respiratory disorder, mixed acid base disorder will be there in acetyl salicylic acid poisoning. Clear? So another condition, ethylene glycol, which by metabolism in our body generate glycolic acid, and basically it is used as antifreeze agent in radiator coolant. So let's suppose somehow a child or someone has accidentally ingested these ethylene glycol. It can lead to ethylene glycol intoxication, which will lead to many problems, and one of the problem is glycolic acid will form glycolate, that is unmeasured anion, and proton will cause metabolic acidosis. So ethylene glycol intoxication can lead to increased anion gap metabolic acidosis, right? Ethanol, alcohol, ethyl alcohol, it can lead to synthesis of formic acid through metabolism in our body, and again it will generate formate ion and proton. You know the whole story up till now. So then another thing, ketoacidosis. You know what is ketoacidosis? Yeah, most of the people know that it is just diabetic ketoacidosis. It has to do something with diabetes. Well, this is not just that. Basically what happens that, you know, our main fuel is glucose. Especially for our brain we need glucose. Other tissues, they can survive on fatty acid, but our brain and I think heart as well, they need glucose. But what if glucose is not present? Glucose is very low. Either glucose is not present, or due to absence or decrease of insulin, glucose is there but cells are not able to metabolize glucose. So either there is functional decrease in glucose, for example insulin deficiency, or actual absolute deficiency of glucose, example starvation. In both these cases, body will not be able to utilize glucose, and especially our brain will not be able to utilize glucose. So it will order our liver to generate some alternate fuel so that it can survive. And what is the alternate fuel? Alternate fuel is ketone bodies. Ketone bodies is the alternate fuel which our brain can utilize, and it will keep working. The problem with ketone bodies is that these are acidic substances and they can generate acidosis. So acetoacetic acid is one of the ketone body which will lead to acetoacetate and H positive. Acetoacetate is, unmeasured anion. Similarly, beta hydroxybutyric acid will generate beta hydroxybutyrate and proton. So ketoacidosis, it can be due to diabetes, that is called diabetic ketoacidosis. Also it can be due to starvation. In starvation, glucose will be very low. So starvation ketoacidosis and diabetic ketoacidosis. Then there is another alcoholic ketoacidosis. I will not discuss that here, otherwise lecture will become too long and then people complain that your lectures are quite extensive, right? So that is ketoacidosis, and ketoacidosis will lead to increased anion gap, metabolic acidosis. Clear up till now? These were the causes of increased anion gap, metabolic acidosis. Let's move forward. So what happens? Let's suppose bicarbonate is decreasing, but unmeasured anions, they are same. There is decrease of bicarbonate without concurrent increase in unmeasured anions, right? In such condition, what will happen? In such condition, anion gap will remain normal. It will not increase. Yes, Dr. Imtiaz, are you understanding? Okay. So again, the same problem, that electrical neutrality won't be there. It will generate positivity. So what will happen? Now unmeasured anion are not here to come to rescue, to increase the negativity, to maintain electrical neutrality, right? These are not here. So what will happen? Yes, what do you think? What will happen? Yeah, so our sleeping chloride. So sleeping chloride will be awakened. And chloride will increase in order to maintain electrical neutrality.
From where does this chloride?
Yeah, good question. She is asking from where this chloride increase. How chloride increases? Basically, the mechanism is not clearly understood. But your book Guyton, it mentions in the chapter of GIT that chloride increased by GIT. From GIT, reabsorption of chloride may increase, right? Or from renal, from nephrons, from kidneys, reabsorption of chloride may be increased. So from GIT, from kidney, also the peripheral cells, they may donate chloride. Chloride will move from intracellular to extracellular to somehow compensate for electrical neutrality. So you caught the point. It is not important here to discuss how chloride is increased, but the concept is that chloride is increased in order to maintain electrical neutrality, and because chloride is increased and unmeasured anions are normal, so anion gap will remain same. Anion gap will remain normal, right? So yeah, so he is saying, today I came to know that positivity is not always good inside our body. There are certain other situations in which positivity is not good. For a pregnancy test may become positive, that may or may not be good for you, depending upon the circumstances. Whether it is planned or not. So of course we are not discussing pregnancy here. So let's move forward, that this type of metabolic acidosis is normal anion gap metabolic acidosis, and it is also called hyperchloremic metabolic acidosis. So yeah, in books you saw that you must have seen that there is written hyperchloremic metabolic acidosis. You must be wondering, what is this hyperchloremic metabolic acidosis? What this chloride has to do with acidosis? So now you are clear about this hyperchloremic metabolic acidosis, this fancy word, right? And the increased anion gap metabolic acidosis, it is also called normochloremic metabolic acidosis, because chloride is not bothered in that situation. Unmeasured anions are there to maintain electrical neutrality, right? So let's move forward. So can you tell me that how bicarbonate can be lost without concurrent increase in unmeasured anions? How can you lose bicarbonate ion? Yes?
Through diarrhea?
Yeah, from two sources, of course. You have two sources, two portals of exit of waste products. Of course, there is sweat also, but mainly there are two. And these are stools and urine. So in the form of, from GIT in the form of stools, or from kidney, right? In the form of, yes? Yeah, either you piss out or you defecate it out, poop out. Yeah, that's it. So from GIT, small intestine, especially small intestine, on the luminal side, it is plastered with large number of bicarbonate ion. You know why? Yeah. Yeah, that's right. So the chyme or acidic rich food, when it comes from stomach into small intestine, it can damage the intestinal lining. Number one reason is that because of it, bicarbonate is there to protect. Secondly, the enzymes, the digestive enzyme, pancreatic enzyme, amylase and all these, they also work optimally at alkaline pH. You know this concept. So in order to maintain the intestinal luminal pH at a alkaline range, lots of bicarbonate is present in the intestinal lumen. So let's suppose you are suffering from diarrhea. So if you are suffering from diarrhea, bicarbonate may be washed out, and you will lead to decrease it will lead to decrease bicarbonate. Yes?
But from where does this
Yeah.
bicarbonate come in the intestine?
Yeah, so she is asking that bicarbonate is lost from intestine. It's a very good and funny question as well, that bicarbonate is being lost from intestine. How it can lead to decrease in bicarbonate in blood? Answer is very simple, because when bicarbonate is lost into stools, it must be replenished, right? Bicarbonate must be replenished. Bicarbonate must come here into intestine to compensate the lost bicarbonate. And from where this bicarbonate will come? It will come from blood. It will come from plasma. So ultimately in plasma, bicarbonate will decrease. And when bicarbonate is decreased, we call this condition as metabolic acidosis, right? So metabolic acidosis will be there, but there is no concurrent increase in unmeasured anion or any other ion, right? So there will be normal anion gap, metabolic acidosis, right? Other route is through kidney. It is little bit complex, so bear with me. So as you know, that we have millions of nephron, and here I am representing a single nephron of your kidney in a much simpler diagram. So from the glomerulus, bicarbonate is filtered out into the Bowman's capsule, and this bicarbonate, it may be lost into urine. But bicarbonate is very important for us. It is our alkali result. It is our acid fighting soldier of our buffer system. We cannot afford to lose bicarbonate. We must somehow reclaim this bicarbonate. We must somehow reabsorb this bicarbonate, and that is done through proximal convoluted tubule of our kidney. So let's suppose this bicarbonate, it was intending to lose itself in urine. So here, this is proximal convoluted tubule. Here we will send a trap. You know, we cannot directly absorb bicarbonate from here inside. Why? Because we don't have special channels that can bring bicarbonate here, right? Are you getting the point? So we have another channel that can throw proton into lumen. So we will use this proton as a trap. So there are special channels. I won't tell you what are those channels right now. So through these channels, proton is sent into lumen, and this proton act as a trap, and it will convert this bicarbonate into H2CO3 carbonic acid. Now another problem, this carbonic acid even cannot be reabsorbed. So this carbonic acid, it must be converted into CO2, which is a lipophilic gas, and this process is catalyzed. This process is catalyzed in the presence of enzyme called carbonic anhydrase, right? Along with CO2, we will also form water, H2O, that I have not mentioned here. So CO2 is small lipophilic gas. It can, by the process of diffusion, it can easily cross membrane, and it will come inside according to concentration gradient, right? Now this CO2, it will be converted back into H2CO3 in the presence of carbonic anhydrase. And this H2CO3, it cannot dissociate into H positive as well as bicarbonate, and this bicarbonate will be reabsorbed in plasma. And H positive, which we initially sent this proton as a trap, we have got it back, and this can again sent out, and this cycle can continue. But with each cycle, what we have got? We have got this bicarbonate, and this is this bicarbonate which we have indirectly recovered, right? So that's how kidney reabsorb bicarbonate. Why I am telling you this process? Because when due to any reason, when kidneys fail to reabsorb bicarbonate ion, you will start to lose bicarbonate ion in urine. So bicarbonate in plasma will decrease, and this will lead to metabolic acidosis, and that is normal anion gap metabolic acidosis, right? So what can be the problem? Let's suppose, acetazolamide, that is a good point, but before that, there are certain conditions, there are certain renal tubular diseases, group of renal tubular diseases, which can lead to, there may be, let's suppose, problem with this channel, or problem with this channel, or problem with nephron. There are different types of that, and in these group of diseases, bicarbonate fail to be reabsorbed, and acidosis occur, and that type of acidosis is called renal tubular acidosis. It in itself is a separate topic. We may deliver a separate lecture on this topic. There are different types, and there are certain pathophysiology. So due to renal tubular acidosis, kidney may fail to reabsorb bicarbonate. One of the reason is this. Another reason is that you are talking about acetazolamide. What is acetazolamide? Acetazolamide is carbonic anhydrase inhibitor. It will inhibit this carbonic anhydrase enzyme, and when this carbonic anhydrase enzyme will be inhibited, what will happen? This process require carbonic anhydrase. It's a crucial step for reabsorption of bicarbonate. So reabsorption of bicarbonate ion will strongly suffer when carbonic anhydrase overdose is there, carbonic anhydrase inhibitor overdose is there, right? So these are the two conditions in which normal anion gap metabolic acidosis can occur. Another thing, this is distal convoluted tubule, and in distal convoluted tubules, there are certain channels through which sodium is reabsorbed from the lumen, sodium is reabsorbed and potassium is secreted, right? And this process happens in the presence of aldosterone. So what aldosterone does is that it increases reabsorption of sodium and causes increased secretion or increased loss of potassium. So let's suppose due to any reason, and we will discuss the reason, aldosterone is deficient. What will happen? Potassium will increase. Potassium in our blood will increase, and that is called hyperkalemia. Hyperkalemia will be there, right? So what will happen? When hyperkalemia will be there, potassium is very important for cardiac contraction, cardiac rhythmicity. Our body cannot afford to increase or decrease potassium, right? So our peripheral cells of different parts of our body, the cells, what they will do? They will pick up the potassium and they will bring this potassium in. They will sacrifice themselves. They will bring the potassium inside in order to somehow lower the plasma potassium, right? But they cannot simply pick up potassium and take it inside, because we don't have such transporters, and also it is against the rule of electrical neutrality. If potassium is going in, some positively charged ion must come out, right? And what is that positively charged ion that will come out of cell? That is proton. So potassium will go in, proton will come out, and this process occurs through potassium hydrogen exchanger. Potassium hydrogen exchanger. This process occurs in GIT, in different cells of our body, as well as in kidneys, right? So because of hyperkalemia, hydrogen will come out and metabolic acidosis will be there. Secondly, because no unmeasured anion is increasing, what will happen? Chloride will increase. So there will be hyperkalemia, there will be hyperchloremia, and because bicarbonate, this proton is there, it will decrease bicarbonate. So it will lead to metabolic acidosis. So there will be hyperkalemia, hyperchloremia, and hypobicarbonatemia. Of course, that is not hypobicarbonatemia. It is called metabolic acidosis. So this type of metabolic acidosis is hyperkalemic, hyperchloremic metabolic acidosis. You know from where this aldosterone come? There are beautiful glands sitting on the kidney. These are suprarenal gland or adrenal gland. From the cortex of adrenal gland, aldosterone is secreted. But what if these adrenal hormone, these adrenal gland, they fail to add up aldosterone in our blood? Adrenal gland, if they fail to add cortical hormones in our blood, this condition is called Addison disease. They fail to add cortical hormones. This is called Addison disease. It is of different types. I won't be discussing the details of Addison disease. But what happens is that cortical hormones are deficient. Cortisol is deficient. Aldosterone is also cortical hormone, that is also deficient. And when aldosterone is deficient, you know what will happen? There will be increased potassium, hyperkalemia, and hyperkalemia will lead to proton, increase in proton. Increase in proton will lead to metabolic acidosis due to decrease in bicarbonate, and then there will be hyperchloremia in order to maintain electrical neutrality. So what will happen in Addison disease? In Addison disease, there will be again normal anion gap, metabolic acidosis, right? Another point. Yes, you have a question? Oh, so he is very fussy about this pink color. So he is saying that has this kidney become pink due to presence of proton? No, it is not like that. It is just an illustration, and it is not anatomically and physically correct. Of course, kidney is not this color. What is the color of kidney? Is it brown color? I don't remember. Okay, whatever. So, of course, by the way, it is not due to the pink color is not due to presence of proton inside kidney, right? So, Addison disease, another condition due to ACE inhibitor toxicity, angiotensin converting enzyme inhibitor toxicity, for example, captopril. So, how ACE inhibitor toxicity can lead to hyperkalemic, hyperchloremic metabolic acidosis? What do you think? Yeah, excellent. So angiotensin 2, it directly causes increased secretion of aldosterone. So angiotensin 2, what it will do is that it will stimulate the adrenal gland to secrete aldosterone, and aldosterone will cause all these actions. But what if angiotensin 2 is decreased, and it is decreased due to ACE inhibitor toxicity? What will happen then? Then aldosterone will decrease. So it is a condition similar to Addison disease, that there will be decrease in aldosterone, right? So Addison disease and ACE inhibitor toxicity will lead to normal anion gap metabolic acidosis, right? So normal anion gap metabolic acidosis causes are, let's recap: diarrhea, renal tubular acidosis, Addison disease, ACE inhibitor toxicity, right? Clear up till now? And increased anion gap metabolic acidosis, this we have already discussed. I won't tell you again, these are just written here, but here another point that I want to highlight here. You know, sulfates and phosphates, they are continuously being synthesized in our body. From where sulfates come, by the way? Sulfur, from where it comes? Main source is what? ATP. ATP has sulfur?
No, phosphate.
Yeah, phosphate come from ATP. What about sulfur? Yes, Dr. Imtiaz, what do you think?
Sugar-based
Metabolism of what?
protein.
No, mainly from cysteine and methionine. These are sulfur containing amino acid. So from protein metabolism of cysteine and methionine, sulfur containing amino acid, sulfate may come in our body, in our blood. Also some sulfates and phosphates, they are directly present, especially in animal diet, as inorganic ions, and in some drugs as well. These are present as minerals directly in, especially in plant based diet, or they may be component of what? Proteins, amino acid, right? Along with that, you know, our body, these are always producing, our cells, they are continuously producing acid, they are continuously producing protons. And one of the reason we are continuously producing proton is, of course, there are different types of proton, different types of acids that we are producing. But one main type of acid that we are producing is carbon dioxide. You know, whenever we metabolize, whenever we oxidize glucose, fats or proteins in order to get energy, we generate CO2. So it means we are producing acid all the time. So protons are being produced all the time, as well as sulfate and phosphate. They are either being produced or they are being ingested. And sulfate and phosphate, these are unmeasured anions, right? Now it's kidney's responsibility to get rid of these unmeasured anions, these sulfate and phosphate, as well as it's kidney's responsibility to get rid of this acid, these protons, right? Now what will happen that if kidney fail to do its task, if kidney failure is there, what will happen? Yeah, it will lead to metabolic acidosis. And not only metabolic acidosis, but these these sulfate and phosphate, these unmeasured anions are also increasing. So this type of metabolic acidosis won't be normal anion gap, but it will be increased anion gap metabolic acidosis. And this is especially significant in chronic renal failure. In acute renal failure, respiratory component and buffer system, they may somehow compensate and they may somehow prevent the metabolic acidosis from occurring. But in chronic renal failure, usually chronic metabolic acidosis may be there, right? Now, this is point that is very important to be understood, that if problem is with renal tubules, if problem is with renal tubules that they fail to reabsorb bicarbonate, that will lead to renal tubular acidosis, and that will lead to increased anion gap metabolic acidosis. But if problem is mainly with the kidney, that kidney failure is there, filtration across the glomerulus, GFR is reduced, then that will lead to increased anion gap metabolic acidosis. So all these concepts, they should be pretty clear in your mind. There is nothing to cram in this. I have seen students, they just cram up these tables, they just all the time they are cramming that causes of increase in anion gap, lactic acidosis, acetylsalicylic poisoning, all the time they are cramming. Do you think you need to cram up these things? These are very logical, right? So you should understand it pretty clearly so you won't forget it again, right? What is the significance of anion gap? One that I have already mentioned, that it is used to classify metabolic acidosis based on anion gap. Based on anion gap, metabolic acidosis is of two types: increased anion gap metabolic acidosis and normal anion gap metabolic acidosis, right? What is the other significance? Quality control of lab results. Well, let's suppose you are a normal person, and I'm sure you won't have any ionic imbalance in your body. Now let's suppose you go to a laboratory and you get yourself checked, and when they calculate different ions, when they measure different ions in your body, in your plasma, and then when they calculate the anion gap from these measured ions, and they find that anion gap is either increased or decreased, but you are normal. Anion gap is, they know that you are normal, you are healthy individual, but anion gap is either increased or decreased. What does that mean? It means that there may be some problem with laboratory. Laboratory equipment is faulty, their methodology is faulty. So it is a crude method of quality control, checking the quality control of lab. These days this is not widely used. There are other methods that are much better than this, right? But because it was mentioned in the literature, so I told you as a significance of anion gap. Clear up till now? Okay, so another thing, that it is used in diagnosis of certain conditions based on anion gap. There are certain ionic imbalances that can lead to increased or decreased anion gap. Some of the condition I have already told you regarding metabolic acidosis. Now some other conditions that you will tell me after looking at this equation. So here is the table, that anion gap may increase or it may decrease. Now it is very logical and it is very easy to understand that in what condition anion gap will increase and in what condition anion gap will decrease, and in what condition anion gap will remain normal. Just by looking at this equation you can tell, right? So we will just look at it one by one. In exam, whenever you see the MCQ, in MCQ you came across any MCQ related with anion gap, you just have to remember this equation in your mind, and then you can easily guess that what condition it will be there, right? In a very logical way. So starting with increased anion gap. So how anion gap can increase? Number one, anion gap can increase when in this equation unmeasured anion increases, right? So what are unmeasured anions? What is the major unmeasured anion? Yes, she is right. So plasma proteins, and major plasma protein is albumin. So in hyperalbuminemia, if albumin is increased in your blood, it will lead to increased anion gap. Clear? So this was about increased anion. Another thing. So what happens that, let's suppose if unmeasured cations decreases, it can also raise anion gap, right? If unmeasured cation decreases, it can lead to increased anion gap, right? What are the unmeasured cations? Yes. Yeah, potassium you can say, but potassium is not much significant here. They just simply ignore it. It is already very small. Yeah, she is right. Calcium and magnesium. So hypocalcemia, decrease in unmeasured cation. Calcium is unmeasured cation. Decreased in unmeasured cation, decrease in calcium. Hypocalcemia as well as hypomagnesemia. It can lead to increased anion gap. Finally, what about these things? What if sodium increase? It will lead to what? Decrease in anion gap or increase in anion gap? What? Yeah, increase anion gap. So hypernatremia can lead to increase anion gap. It is very easy to understand, right? Now let's talk about decreased anion gap. Hypoalbuminemia, talking about unmeasured anion. If unmeasured anion decreases, unmeasured cations are same. Unmeasured anions is decreased, so anion gap will decrease, right? Clear? Hypoalbuminemia will lead to decreased anion gap. What else? If unmeasured cations, they should increase or decrease? So they should increase, and this is normal. They should increase in order to decrease anion gap, because minus is here, right? Are you confused or you are with me? Okay, good. So what are unmeasured cations that may increase? Yes. Calcium, hypercalcemia, hypermagnesemia, and other condition. Can you? Yeah. Hyponatremia is, yes, we will talk about that. What about cation? Another cation, can you tell? Have you heard of a drug, antipsychotic drug, which is rarely used these days? Lithium. And lithium is, it is an ion, it is a charged substance. What is its charge? Lithium. Lithium is positive charge. So what if there is lithium toxicity? It is positive charge, it is cation, and it is unmeasured cation. So if lithium is unmeasured cation, if lithium toxicity is there, it can lead to decreased anion gap. Clear? So it's very easy and logical to understand. Just by looking at this equation, you can solve each and every possible question, MCQ or even SCQ, related with anion gap. Right? Now, what if sodium decreases? So if sodium decreases, anion gap will decrease. So I will talk about this later. So hyponatremia. There is another condition called hypergammaglobulinemia. What are gamma globins? Have you heard of gamma globins? What is that? Yeah, antibodies are major gamma globins. There are some other gamma globulins. Yeah, you are right. Gamma globulins, not globins. Gamma globulins. My friend is very much fussy about accent and pronunciation. Thank you about that. So, hypergammaglobulinemia. If gamma globin increases due to any condition, it will lead to decreased anion gap. Why? Can you tell me why? Because not all plasma proteins are negatively charged. some plasma proteins are positively charged, and gamma globulin are those plasma proteins that are positively charged, that are cations, and these are cations which are unmeasured cations, right? These are unmeasured cations, and because of increased in unmeasured cations, anion gap will decrease, right? Any other condition that is not mentioned here, can you tell me? It is very easy to understand. If you are taking any drug that is positively charged, for example, I told you about lithium, another drug is polymyxin. Polymyxins are antibodies, and not antibodies, antibiotics. Antifungal? These are antibiotics, I think. So polymyxins are the antibiotics. These are drugs that are positively charged, and because these are positively charged, they will lead to increased unmeasured cation, which means decreased in anion gap. Then in metabolic alkalosis, anion gap is increased. Why this happens? No, I am not gonna tell you the answer to this question. You have to find the answer. Of course, I know the answer. Don't think that I don't know the answer. So I know the answer, but you have to tell me the answer in comment section of this video, right? So this was about everything that is possible to explain in this short, somewhat short video of anion gap. Of course, this is not that much short. So I cannot tell you each and every possible cause of increase, decrease, or normal anion gap. These are that you mainly encounter in your exam. But up till now, if you have clearly understood all these things, if any cause it comes outside of this lecture, you can easily guess it out, right? Now, what you need to remember in this anion gap, what is important from exam point of view especially, the only thing you must know is this equation, number 1. And if you know this equation, second most important thing is that you must know its significance, its causes, that in what situation anion gap is increased or decreased, or in what metabolic acidosis anion gap is normal or anion gap is increased. That is just that you have to remember. All this explanation, it was just to give you a complete concept of this, in order to, that's what I call demystify the concept of anion gap, right? So if you like this video, please subscribe to my channel, please like this video, please share your precious comments, and please join my group. Here in that group you can also find the notes of this lecture, and in further lecture I will also share the notes of upcoming lecture in that group, right? Thank you so much for watching this lecture. Goodbye.